The first thing about DNA structure that we will be discussing is base pairing. As you can see here, an A is paired with a T and a G is paired with a C. An A is a purine and a T is a pyridine, and a G is a purine and a C is a pyridine. So these are paired together because they are both reverse complements of each other. Next thing highlighted is the hydrogen bonds. A and T is connected through 2 hydrogen bonds. G and C is connected through 3 hydrogen bonds. These are weak attractions between positive and negative charged molecules. The phosphodiester bond is created by the connection of the 5' phosphate group to the 3' OH group of adjacent nucleotides. These combine together all along one of the DNA strands to form phosphate backbone. Base stacking occurs when the flat base pairs are perpendicular to the sugar phosphate backbone, which then creates the maximum hydrophobic interactions and stabilizes the double helix. The difference between ribose and deoxyribose is highlighted here. As you can see, the only difference is in the second carbon group, where there is an OH in ribose and deoxyribose has H. This difference makes it possible for the enzymes involved to distinguish between the two sugar molecules. Otherwise, they are the same.
This is highlighting the difference between the major and minor groove in the double helix of DNA. The major groove is on one side of the helix and are further apart, and the minor groove is on the other side and shows that the strands are closer together. This also shows this piece of DNA is right-handed because, as you can see on the front strand of DNA, it is going up and to the right.
Next, we are going to look at different articles or advertisements or news posts that show left-handed DNA, which is the more rare type. So this one shows left-handed, and you can tell because the upper strand is going up and to the left direction. So this DNA, you kind of have to turn to the side a little bit, but once you flip it right side up, you can see that the DNA top strand is going up and to the left direction again. Here's another one where how it's pictured might be kind of difficult, but once you flip it upright, you can see that that forward strand is going up and to the left.
So this is a very small segment of DNA, but when you look at it, there's just that one crossover, and it is going up and to the left when you turn it around a little bit.
So this is the last one we're going to be looking at, and this one shows the DNA being used as a jump rope, but that front strand is going up and crossing over to the left.